ahora para introducir al conferenciante le voy a dar la palabra al profesor de la Facultad de Ciencia y Tecnología del Departamento de Ingeniería Química, Gorka Elordi. ¿Mesedez, Gorka?
Bueno, kaixo guztioi, lehenik eta behin eskerrak etortzeagatik. Duela hilabete batzuk, Fernandok aipatu zidan, ea algenuen zerbait antolatu asteazken kulturaletako egun hauetarako, nolabait bioteknologian oinarritua. Alde batetik mundu guztiarentzako izan behar da, hori ikasle guztia, fakultateko ikasle guztiontzako, baina bestetik baita pixkat zentratuago bioteknologian. Eta kasu honetan, nire helburua zen denontzako, baina baita ere nolabait bioteknologia aplikatua. Bakita asko gustatzen zaizuela oinarrizkoa eta baina nolabait kombinatzen dituena bi arloak, ezta? Eta nik uste dut Kail horretarako aukera oso ona izan dela ona etortzea eta benetan ondo doitzen dela nahi genuerako. Nik uste dut hori ikusi ahal izango duzuela. Bera, Kaust Unibertsitatean ari da, ikusten duzuen bezala, Algal biotechnology, up and downstream engineering of green cell factories izango da hitzaldia, eta aldi berean hitz egingo du baita nola modifikazioak eta genetikoak eta baina baita gero bioprozesuaren diseinuak eta hobekuntzak. Eta bera Kaust Unibertsitatean egiten du lan, eta hainbat argitarapen dauzka, bai bioteknologian, mikrobiologian, ingenieritza kimikoan eta horregatik uste dut, bere ikuspegi zabala egokia izango dela egun honetarako. Como os decía, en primer lugar, gracias por venir. Y Fernando me comentó para que organizásemos una charla de los miércoles culturales, más o menos centrada en la biotecnología. Y aunque yo no soy experto en el tema, estuve intentando buscar algo que nos encajase, por un lado, para todos los alumnos o toda la gente que puede estar interesada, pero por otro lado también un poco más específico. Y en este caso, mi objetivo era buscar a alguien que podría combinar tanto la parte fundamental de la biotecnología, modificaciones genéticas, etcétera, también con la parte aplicada del diseño de procesos, y creo que Kyle en ese aspecto es una persona adecuada. Él nos va a hablar de, la conferencia va a ser de Algal Biotechnology, Up and Downstream Engineering of Green Cell Factories, y como os decía, él tiene publicaciones recientes en revistas de gran impacto, tanto en biotecnología, en microbiología, en bioquímica, como ingeniería química y química, y por eso creo que es un muy buen conferenciante para toda esta comunidad. Y sin más, ya le voy a dar paso. I just introduce you briefly, so just the audience is yours, okay?
Okay, good morning, everyone. Good afternoon, I guess. I don't know if that was a nice introduction, but I think it was. So thank you very much, Gorka and Fernando, for the lovely hospitality and to be here today. I'm going to try and give you some entertainment and education at the same time. We call that edutainment in English. I hope you stay awake for a whole hour. They told me to give you a lecture as if you were one of my students in our hour-and-a-half-long classes, but I'll try and keep it only 1 hour. These are some photos that represent the kind of things that we do in our lab and the view from our cafeteria. So I think you have an equally nice view here. Ours is of the Red Sea, yours is of green fields, and probably not, you couldn't probably have any more different of environments, but we can be bound together by one nice biological phenomena, and that is photosynthesis. So I'm hoping that, yes, the slides are changing, this is great. I need to tell you a little bit about my lab. We are called the Sustainable and Synthetic Biotechnology Group. In English, we say this is a mouthful. It's a lot of words on one slide, and what does it mean? It means that we work with all kinds of biotechnology that involves somehow the recovery of nutrients that are lost to the world. So somehow resource use recovery. And we do this with algae as one of our main workhorses, but we're not limited to algae. There's a lot of bacteria that do photosynthesis. There's a lot of waste products that can be grown heterotrophically in different processes. So algae are not the only thing we do, but they're the major thing we do. And being the only algal lab in the Arabian Peninsula, we have to do a little bit of everything. So we call ourselves specialized generalists. We find new algal strains, just like Professor Sergio's doing here. We have our own library bank. We try and engineer those strains to do things that they don't normally do. But once we engineer them, we actually need special bioreactors in which to grow them and get those chemicals out that we've engineered in. And we try and talk about how can we scale these processes. So that's these 4 bullet points here. Bioprospecting and harnessing is making a cell library. Microbial cell factories is turning those cells into some kind of bioproduction chassis. And bioprocess design is everything that comes after you've designed your cells. So how do you grow the cells? How do you get that product out? And to reiterate what I think Gorka said he was going to say, the downstream processing is much harder than the bioengineering of the cell. It's 80% of the cost of any bioprocess, and it's really where you need to put a lot of your efforts. So think about that when you're studying. It's not just about modifying the cells, doing CRISPR or whatever. It's what do you do after that to get the value out. And so we also talk about resource cycling, which is where we build the outdoor cultivation facilities to do this. A little bit about where I'm from. So Pedro Castaño is a professor that connected us. He's from here, so you've heard about KAUST before. It's a small university only for master's and PhD students. We have about 1,500 students, about 200 faculty, and there's about 8,000 people that live in our small city, which is like if you took your whole university, plopped it on the side of the sea and put a wall around it and said that's a city. So we have a nice place to live. It's kind of like living at a tropical resort, lots of palm trees, and all we do is science all of the time. We're here on the coast of the Red Sea. It's a rather unusual geography for you, I would assume, but it's actually quite a nice place to live, and we can talk more about that after if you want. I think your preconceived notions of what the Arabian Peninsula look like may be something like this. You may have these images in your mind. And it's true, these are photos I've taken around the kingdom. It's a dry place, it's an extreme place. There's hot temperatures, very little fresh water. But with a little bit of management, it can look like my backyard. And the only difference between my backyard and the desert is that the wastewater that's processed on campus is recycled into our irrigation system. So all of a sudden we have water and hot temperatures and photosynthesis. It turns out plants like this and algae like it as well. It's not that it's prohibitively hot, it's just that we need to manage our water a little bit better. And so we look at the kingdom in general as a place that can have all of the right conditions for algal biotechnology. So we have lots of unused land, we have access to seawater on the coasts, and anywhere human beings do what human beings do, we produce a lot of nitrogen and phosphorus wastewaters. And that is actually the substrate that algae grow on the most. So KAUST has these 4 foci as its scientific themes: food, water, energy, and environment. And we're also starting to get into health and digital technologies. We have a big AI push now as well. So the kingdom is interesting for this, and the idea is that we want to install algal biotechnology in the kingdom and use local resources. These are examples of algal photobioreactors from around the world. You have some, this big tubular system here is in Portugal. The hanging bag system is in northern Germany. And this big pond here is in Rio Grande do Sul in Brazil, in southern Brazil. They can be installed anywhere. It doesn't have to be in these geographies. They're just different ways of getting photons into a liquid solution to drive photosynthesis and allow algae to double and use that energy to convert waste nitrogen, phosphorus, with some carbon dioxide into their biomass. Algae are primary producers. We know this from fundamental biology, fundamental phycology, but they can also be primary waste stream conversion vehicles. At the end of the day, what algae look like when we harvest them is a powder that's not that much different from flour when you buy it for baking, or the yeast you would buy to make bread with. It's just that it has different colors. So algal biomass, once it's processed from these bioreactors, is a nice freeze-dried powder or spray-dried powder that you can then extract chemicals out of. You can use it for a lot of different things. It has a lot of value chemically. So what we've done is we've taken waste inputs and turned them into higher chemical order value. That's really what algae are all about, higher chemical order value conversion. These are different biomasses from different species, and you can buy these. You can buy these commercially in kilo bags. They're quite expensive, but you can buy them. And the idea is that each species has a different unique biochemical composition. That's visually shown here in their pigmentation. So a lot of these species you'll hear Professor Sergio talk about from his collection. We have Hematococcus, which is producing the red pigment that accumulates in shrimp, flamingos, and salmon. We'll get more into that in a bit. But one of the things I like to point out here is that Chlorella, Dunaliella, and Hematococcus, which are these ones here, Chlorella, see green, orange, and it looks brown here, but it's actually kind of a nice red. These are all green algae by phylogenetics. They're green algae that the only difference is their pigment composition. And we ascribe them to the green algae based on their evolutionary history, not necessarily their coloration. So just with a couple gene differences, you can have massive differences in your biochemical composition. Now, in the kingdom, the ministries are investing very heavily in this kind of technology, and we're building larger and larger places to capture sunlight, convert wastewater into algal biomass. And the idea here is to couple it with local shrimp production, local fish farming, so that we can have the literal nitrogen and phosphorus from shrimp and fish poop water turning into algal biomass that can then be a high protein food source to go back to the shrimp and fish. Because we don't want to be importing a lot of feed. It's very carbon intensive, very, very expensive. So we have a small production facility, which is just about the size of this stage, times 2. And that we can do some small test beds, but they're building currently a 4.2 hectare facility also for this process. This is not my lab, this is not what we're doing, but what we are doing is supporting them. And much like what Professor Sergio is doing, we're making our own local library of algae strains. So we call this the Algal Living Library. And the idea here is that since about 2014, there's something called the Nagoya Protocol, which means that if I go to the Brazilian rainforest or Bilbao and I find something that's biologically really exciting and I bring it home and I start making money with that, the government of that place can sue me for doing so. And so every place in the world should be developing its own local resources for these sort of things. And we're no different. We're also no different because our environment is much more extreme than yours, and the algae we isolate can tolerate different temperatures, light, salinity, that sort of thing. So far we've sampled on all of these places up and down the coast. We'll be going to the other coast at the end of the month, and we're continually doing this. It's an ongoing project for the last couple of years. We have about 80 species that we've isolated as monocultures. They're not completely pure, they may have some bacteria, but we call them monocultures because when you grow them, that's the most prominent organism there. And we're trying to identify what they are. Just you can do it fundamentally by the microscope, but that only tells you so much. A lot of green algae sort of look the same. So is that green algae 1, green algae 2? We don't know. There's where genetics comes in. This is really powerful, but what we've done is also invested in a lot of infrastructure to then characterize these strains as well. So you can isolate an alga, but once you have it, it's a lot of work to actually figure out what value it is. We're very fortunate in that we have the largest suite of photobioreactors for a lab of any research lab in the world. We have 32 of these independent reactors, which means that we can individually grow under different environmental conditions in multiple different conditions at one time. And we program them with local weather station data. And what we do is any new strain that we isolate, we then take and we grow in a modeled spring, summer, winter, fall, and control cultures. And that gives us 6 reactors to say, oh, you know what? If we want to scale that up, it's probably not a good idea to do it in February. Or, oh, that strain actually does really well in August. So this is really important information that we're collecting for the kingdom to build its own bioresources. If you're interested in looking at any of the species, this QR code will take you to the library. You can download it. We update it every couple of weeks with whatever new data we have. You won't be able to get any of the strains here, but it's interesting. Maybe it influences some of the work that you're doing, especially I'm seeing some photos here, so that's good. Just wait until we're done. We're good? Yeah, got it? Awesome. Why is this important? It's important not only to understand how these cells grow, but to have little samples of algal biomass. And most of an algal culture is water. It's not particularly dense when you grow it under these conditions. So from these 400 ml reactors, we can get little baggies. They look kind of like drugs, don't tell anybody. We can get each one of these little baggies, and that's enough to tell us how much protein, how much oil, how much pigment each one of these strains has. Down here are the ones that I used for the photo of all the powders. So you can see the difference in scale between what you can buy commercially and what you can grow in your lab. So you get a lot from a little with this, but it's the first time that anybody's tried to do this in our geography. We're also very fortunate that we have our small outdoor production facility, and they've already been able to take strains that we've isolated and scale them up to several thousands of liters. So just so that you understand, when you grow algae, you get about maximum 2 grams per liter of biomass. So in one of these 1,000-liter reactors, you can get about 1 kilo of biomass in a week. So 1 kilo is not that much. Like this laptop weighs almost 1 kilo, right? It's not that much biomass. But if you produce continuously over and over and over again, and your inputs are poop water or some kind of wasted industrial process water, now all of a sudden it's free, high-value chemicals that are coming out of something that otherwise would have been thrown away or we would have had to pay away to throw away. What I was really surprised about is that this became sort of national news in our region. I was not consulted on any of these articles, so some of it is not entirely true, but it's still kind of nice to see your face show up in random articles in different newspapers, wherever you are. Oops, that's the wrong button here. There we go. Okay, so to summarize this introduction part, algae are really good at converting waste into higher chemical order value. They're not particularly great at sequestering carbon, and I've spoken to some of you about that earlier. They do use carbon dioxide as their main source of producing their biomass. But if you think about the numbers that I said, 1 kilo per 1,000 liters per week. In algal biomass, just to give you some numbers, you can estimate 1 kilo of biomass is 2 kilos of CO2. And the reason for that is CO2 is 33% carbon. Biomass is about 50% carbon. And so you round it up, it's 1.82. You can say 2 kilos of CO2 go into 1 kilo of biomass. We as human beings emit carbon dioxide in the megaton amounts. And so the amount that you're putting into your biomass is not that high. So at the end of the day, what we're really talking about doing is converting waste nitrogen, phosphorus, and some carbon dioxide into a higher chemical order value. Now, my whole shtick since I started being a PhD student, which, you know, I was in your position not that long ago. I realized I defended my thesis next month, 9 years ago. So it's been quick, right? It's been quick to transition into professorship. So you could be where I am someday not that far in the future. My whole thing has been to say, okay, well, if we have this relatively inefficient system that makes a nice biomass that is cultured on waste, can we engineer it to produce other chemicals that might be even higher value, or just make the economics of that situation a little bit better? So the background here, and this is true for all metabolic engineering, which is the idea of taking a pathway from an organism that produces something of value and putting it into an easier to handle organism. Classic example is a small plant in the Brazilian rainforest makes an anti-cancer drug, but it doesn't grow very fast and it only grows in that specific place, so we can't farm it, and we can't farm it fast enough to get enough of this anti-cancer drug. But with genome sequencing, you can find out the genes in that pathway, and it turns out that most metabolism to make specialty products is modular. So you'll have some kind of precursor that's a common intermediate in carbon metabolism. A few genes later, maybe it's up to 20 genes later, you can get to that specialty product. So you can, through metabolic engineering and iterative cycling, add value to a microbe and make it that same product that you would have made from harvesting that native plant in that microbe. And we do the same thing in algae, and we do it in this iterative, what we call design, build, test, learn cycle, which you'll hear if you are a synthetic biologist and care about buzzwords. It's much more difficult to do this in algae because their genetics are really complicated, and their genetics are complicated because they don't like changing. So algae in general are algae because they have not evolved to more complex organisms, and they do that by keeping their genetics very, very tight. So when we talk about algae as a concept, we look at it from a bioengineering space as this multi-dimensional space. You have your flask of algae. It's producing oxygen. We saw a poster outside about producing hydrogen under the right conditions. But really what you have is a biomass and an aqueous phase. And that biomass has soluble and insoluble phase. You can also engineer volatile chemicals to come out of that biomass, whether they're hydrocarbons or more hydrogen. The extracellular aqueous phase is actually not empty. It's full of proteins that the cells secrete, cell walls, that sort of thing. Those can be engineered. And you can also engineer specialty chemicals that then need to be milked from the cell. So milking is where you interact the cells with the solvent. And that's what's shown here. I'll get into more detail in that in a second. So since 2014, we've been very busy on doing this, specifically with green algae, and we've produced a lot of different chemicals, a lot of specialty chemicals, C20 medicines, C15 perfume products, different platform chemicals, things like isoprene, the monomer that's used to make rubber. We have modified lipids, and we secrete recombinant proteins. We also have changed the pigment composition of both red and green algae to go from looking green to looking sort of browny red, and I'll talk a little bit about that in 1 second. But just to tell you about the organism we work with, so there are some posters outside on Chlamydomonas reinhardtii. This is a model green alga. It's been in the lab since 1940, and it's just really easy to handle. You can grow it on a benchtop with a little bit of light, does really well. You can transform it, and you'll get colonies back in a week. It's very easy to handle, but it's also very difficult to work with genetically. The nice thing about working with model organisms is that they're highly domesticated. And when you work with a wild type alga, oh, sorry, you didn't even see any of that stuff. I'll go back 1 second. There you go. That's what I just said. Okay, we're keeping on, it's easy to grow, highly domesticated. I have 2 different screens here, sorry. This is the right one. I should look at this. So when you isolate a new alga, and I'm sure Sergio's team will be able to tell you this, they don't want to work for you. You have to trick them into working for you, just like original corn that was isolated was not very tasty. It had to be domesticated. And so when you have a wild type alga, it may have a colony formation that's dry, and you can't pick it with a toothpick. But if you get rid of the cell wall by chemical mutagenesis, all of a sudden it's goopy, and it can be picked with a toothpick or a robot. This enables us to handle algae in a way that allows us to do bioengineering, so high throughput. You want to make sure they don't stick to plastic so that you can grow them in microtiter plates, for example. This is very, very important. You also want to be able to pick with robots to do higher throughput, and that's what this, this is my handiwork, listening to an audiobook and using toothpicks, and then that's a robot picking. Clearly, the robot's better at it than I am. We're now in a phase where we're able to work with engineering algae because DNA synthesis has become affordable. This was not always the case. Even 10 years ago, this was way too expensive to do regularly. But now what we can do is design in a computer the DNA sequence we need that tricks the alga into thinking that that gene is its own, and we can have it printed. And this is really powerful. So now you're hearing about completely synthetic E. coli genomes. The yeast 2.0 genome papers came out 2 weeks ago where they've been building completely synthetic chromosomes. This is going to be the future of algal engineering. You take some property of the cell that you like, you use that cell to do whatever bioconversion you want, and you re-engineer its genome completely. We've made molecular toolkits for Chlamydomonas. I'm not going to go into any of these details, but there are different molecular toolkits out there. There's styles like MoClo, which is using this thing called Golden Gate assembly, where you can assemble many parts of DNA all at once. And then there's more brutish-like plasmids like POP3 that I made, where you can open and close them anywhere you want to drop in a gene. The tools are different for different reasons. MoClo is fantastic if you have a lot of different parts that you want to multiplex and try at the same time, because the physical act of building those plasmids is faster. But if you have 1,000 genes that you want to tag with YFP, you can do it much faster with the p-opt kit. So these don't really matter, and if you're not a biologist, don't worry about it. Just like you have a hammer and a screwdriver in your toolkit, we have different plasmids that allow us to modify the genome. Now, why is it so difficult to work with algae genetically? In general, up until the mid-2000s, so 2005, 2006, it was known that you could transform and get colonies back on a selection plate. So you could get antibiotic resistance, and you could get like a little bit of GFP expression. But here's the problem. GFP, you can see even if there's a very little amount of expression. And antibiotic resistance functions by enzymes that are able to chew through a lot of antibiotic, even with a very low level of expression. But if you want to do metabolic engineering, you need really strong overexpression. And those genes are much larger than reporters or antibiotic selection markers. So the nucleotide footprint, as we call it, is much bigger. And it turns out that that just didn't really work at all in green algae. Now, I'm not going to bore you with 5 years of how we figured this out, but we were very lucky that we figured out a way to trick the cell into thinking that gene was its own. And in eukaryotic genes, there are these regions called introns. They're not coding, and they're removed from a sequence as it's becoming a protein. And it turns out that in green algae, if you don't have these introns, you don't get any expression. And what's illustrated here is that when we build synthetic sequences that have introns every certain number of base pairs, once we get to a place where there's the right pattern, in this case it's with 3 introns and a 4th bigger one, then the cell thinks, oh, this is my gene, I'm going to express it. But if we have any one of those missing, the expression goes away. And so there were generations of grad students, because genetic engineering has been around since the 1980s, 1990s. There were generations of grad students who burned themselves out trying to make chlamydomonas work as an expression host, and it never worked. So we were lucky enough to figure this out, and all of a sudden, overnight, we went from not being able to do anything to being able to do almost anything we wanted with this cell. And that really changed the game in terms of our ability to do biotechnology. Just because there are probably a lot of biotechnologists here, if you want to be considered important in your field, you have to build tools that other people can use. And so biologists in general are very lazy people, myself included. Keep that in mind, biologists are lazy, you can quote me on that. It's easier to figure this stuff out if somebody makes a server for you. So what we did is we coded a server where you can put in any protein you want to express, and it will automatically give you this optimized sequence that has these elements in it in the right pattern, and then you can drop it into the toolkit that we made, and it will express in our organism. So we built this back in, 2019 it was published, so we built it back in 2017. It's been very, very popular in our field. So the lesson to the young students out there, think about things that you can build that other people can use, and you will become known for it. It's a very good life strategy. So this tool is out there. There's another reason why green algae are difficult to engineer, and that's because they don't allow targeted integration of the gene as it's going into the genome. So when you throw in a piece of DNA, it randomly goes in. And you can imagine in a 3-dimensional genome that there's some complexity happening there. So you have this phenomena of non-homologous end joining. The DNA goes into the genome, and then it's expressed better or worse depending on where it goes in. So if you just express YFP and you pick 96 colonies, you get about 50% expression. But if you want to express a metabolic enzyme like this bisaboline synthase, you get 1 out of 100 that actually expresses very well. So we end up having a numbers game where we really have to use modern technology to overcome that. And so we're very fortunate, again, not only to have a lot of bioreactors, but we have robots too. And not just grad students. Grad students, that's not an insult to grad students. Those are also robots in some people's minds. But these robots allow us to go much more high throughput. So now we can allow picking of colonies at 384 density. We can stamp them between one plate and another. And that allows us to go through iterative cycles, design, build, test, implement, learn with our alga much more rapidly and more efficiently. So we can do about 10-fold faster colony picking with a robot, and there's no human error anymore. A normal engineering workflow of algae looks something like this. You'll start, you put your DNA into a flask with some algal cells and you vortex it, and it opens up the cell and the DNA goes in, and then you need to find the cells that have the DNA in them. And so for that you use agar selection. You can see plates dying over here on some kind of antibiotic. Then colonies come back. Those are the few out of a billion cells that got the DNA that you put in there. You use the robot then to pick them, organize them, and you get more and more organized until you can do some kind of screening. And for us, what we realized is that fluorescent proteins are the easiest thing to screen in algae. So you may notice there's some black plates over here. And what we've done is we've mixed a chemical called amido black. It's a protein stain. We mix that into our plates, and all of a sudden the background fluorescence goes away. So this was a little hack that we had to come up with in order to work with the cells, because you need this ability to screen for fluorescence. So these colonies that are shown here, they're a very faint reporter. They're orange, hard to see normally. If you shine blue light on the plate and look in the orange spectrum, you get all this background interference. But we can do this much more easily with our engineered strains. So the DNA is difficult to design. You require synthetic DNA. You have to adapt to the cell, and it's a numbers game. And we can improve this by robotics. So it's quite challenging, right? And one of the things that we always say to our students is you have to come up with creative solutions. So photosynthetic cells are, in their own way, fluorescent. If you shine blue or red light onto a photosynthetic tissue, it will emit red light back. And it's been talked about for years in literature that this is somehow inhibitory of doing fluorescence imaging. Here you can see colonies lined up on that blue plate in white light, and they're barely visible up there as little dots, but underneath you see the red fluorescence. And what we did is we said, well, if we want to work with a lot of fluorescent reporters that have different excitation and emission spectrums, we need to be able to screen them just as efficiently. So it turns out that if you use very, very narrow bandpass filters, you can actually isolate those individual fluorophores from one another, and you can see all the way up the spectrum, even in a photosynthetic cell that's emitting all of this extra red light. So this was something that we did just as a technical hack to allow fundamental investigations in genetic engineering, right? It's not particularly scientifically novel or engineeringly complicated, but nobody had gone through and purchased a bunch of little filters and tried them to actually make this work. So there's a lot of areas where you can think about optimizing what you're doing in the lab that may not necessarily involve all that much difficulty. It's just being the first to do it, right? So here you can see it's very difficult to see here, but from the blue all the way to the far red, we can image on the plate level this way. Okay, domestication. One of the things that algae do is they grow in nitrogen and phosphorus containing liquids, but so do plants, so do fungi. A lot of things grow in that situation. And one of the ways you can overcome this is by engineering cells to use alternative inputs instead of the classical inputs that they normally would have. One of the things we did to domesticate our cells is engineer them to grow on phosphite instead of phosphate. So phosphite is PO3 instead of PO4, and it's non-bioavailable. It comes into the cell, but the cell can't use it, and they eventually starve because they can't get enough phosphorus into them. So this gene, this PTXD, it's an oxidoreductase. It allows the cells to make phosphorus from phosphate from phosphite. The other thing you have to consider, and this is where biotechnology students need to be very aware, the cultivation of your cells is highly affected by their fundamental metabolism. And one of the things that you'll learn is that if you feed too much ammonium, you get pH issues, because as ammonium as a nitrogen source is consumed, it releases a proton. So the more ammonium you have in a liquid source and cells are eating it, the more the pH will crash. And so using nitrate allows you to jam more nitrogen into a culture vessel. And these are things we didn't understand when we started working with algae. We just had some kind of lab medium that we used, and we grew the cells, and we said, oh, they don't grow very well. But then we talked to the bioengineers who said, yeah, they're not growing well because your pH is crashing on day 3. Well, what if they could grow on a different nitrogen source? Oh, it turns out that if we put these two plasmids in, we can get them to grow on nitrate. Isn't that convenient? Now all of a sudden we don't have crashing of our culture, but we have these nice dense green cultures where instead of getting 1 gram per liter, we can get 20 grams per liter biomass because we can add more nitrogen, they can build more proteins and that sort of thing. And then you can use these kind of cells to produce different products. I'm not going to go into the details of this, but the important thing is by modifying your nutrients like this, you can get very high density cultures. So in the next slide, I'm going to show you a high density culture. But what's really important is that we were able to show that the cells could use the same nutrients, but with phosphite instead of phosphate. So in this case, we have a non-bioavailable source of phosphorus. We have a high density medium with a lot of nitrogen in it. And the cells grow exactly the same that are engineered to use this phosphite. That's not so important to you right now looking at these growth curves. I mean, this is really boring. But what it means to the bioengineer is that when you don't sterilize your medium, you don't get contamination. So on top here, you see fungus that's in non-sterilized medium when we grow the algal cells that just takes over the culture and the culture crashes. And that's what this disgusting white goo here is. But if you use phosphite instead of phosphate, nothing else can grow. And then you have no contamination in these bottom plates here. Okay, I told you we engineer cells to do chemical production. And a lot of the chemical production we're trying to do is from natural products from plants. One of the biggest issues that you face when you start making new chemicals into a biomass is that these chemicals have different hydrophobicities. A lot of natural products are oily. A lot of natural products are not hydrophilic. And that's challenging in a microbial cell because they have nowhere to put these products. So you can think of a plant. If you touch a mint leaf or a sage leaf, your fingers end up smelling like sage or mint. That's because you're breaking these little hairs that are on the surface of that plant. They're called trichomes. And they are specialized tissues that are full of oil that are hydrophobic. So natural products can accumulate there. Microbes don't have that structure. And so one of the ways that we overcome this is we use a culture solvent interface. So you need a biocompatible solvent. In this case, it's dodecane is used, and it's a light alkane solvent. It's twice the size of hexane, which is C6. It's C12 dodecane, and it floats on top of the culture. It's great in the lab. You can do all kinds of high throughput screening, but if you try and put it into a big bioreactor, the hydrophobic products from the cells start mixing with the solvent, and you get a foam formation and emulsion, and this ends up blowing out of the reactor in the off gas. It's also highly flammable and explosive, which is not great if you're thinking about a few thousand liters of culture. So one of the things we had to do is try and figure out, how do we build a bioreactor that allows us to milk these chemicals that we're producing from our algae? So in this paper, we explored hollow fiber membranes. These are usually used in wastewater treatment to have high surface area where you can have water going through in a very narrow pore in high little straws that are actually just like a membrane but straw. And then you can filter out microbes from wastewater, or you can filter out chemicals from wastewater. But what we did is we kept the solvent on the inside of the straws and the culture on the outside. And that allowed us to have a high surface area contact between the algal culture and the solvent, and we were able to continuously milk away these products. This was done in small scale, but it ended up getting published very high impact, so I was happy about that. And there was a lot of bioprocess computation that we did as well. But we think about solvents as being lighter than water, but bubbles are always going to come out of the reactor, and that's always going to be at the surface of the culture and the solvent. So wouldn't it be nice if we had solvents that were heavier than water, more dense than water? And that's actually what we were able to figure out. So there are these class of solvents that are called fluorinated, perfluorinated solvents. They're of many different chemical structures, and they're more dense than water because they have fluorine groups. So these perfluorinated solvents sit underneath your culture, which means that you can bubble as much as you want, and it doesn't disturb the interface. And so from this, what we were able to do is come up with a few new cultivation styles. We could show that terpenes were actually able to go into these products, these chemicals that we were engineering, could go into the solvents. And that allows us to come up with some different bioreactor designs. I'll show you one in a second. But just to show you that, just what's in textbooks may not always be the best thing. When you use dodecane, it's a petroleum extract. It's a petroleum distillate, right? It's purified from other petroleum products. It's not very clean. And you can see that here in this gas chromatograph. So the product we're looking for is that peak in the middle, patchoulol, but there's all these other peaks in that dodecane. Whereas with our fluorocarbon solvent, there's almost nothing. It's a very pure chemical. That means that our natural product that we're making is not contaminated with petroleum distillate. It's a much nicer way of getting our product out. What's even nicer about this is that the fluorocarbon is so dense, it doesn't mix with other solvents. So in this case, we're talking about a perfume, patchoulol, and patchoulol, it's soluble in things that perfumes are soluble in. When you buy a perfume, it's usually got some ethanol in there. Well, it turns out that if you mix the fluorocarbon containing patchoulol with ethanol, all of the patchoulol goes into the ethanol. And so we not only have a nice solvent extraction from the cells, but we have a super easy way to get that product that we want into something that's usable. So all of a sudden, it's directly usable in ethanol with 2 steps. So from a bioprocess standpoint, that's really great. citric acid, the stuff that you use to make sour candies, takes 14 steps to make, for example. So this is a big win for us. Like I said, this made us come up with some crazy ideas for different bioreactor styles. We're building some of them now, but the idea is to create a roll-to-roll process, or a liquid-liquid extraction, where all of a sudden you can have an algal culture there and a perfume product coming out on the other side. So just to see if you're awake, I actually brought some perfume for you. Let me see if I can find it here. It's deep. There we go. There's one. I'll give them to Gorka and he can pass them out. This is a free COVID test for everybody too. This side, another one. You have to open it. It smells much better if you rub it on your wrist. So there'll be a note of ethanol because it's suspended in ethanol, but then there's a lot of perfume products that come out after it. This is a great way to keep everybody awake. So while those are going around, you can just feel free to keep passing them through the audience. I'm going to tell you about another perfume story that led to the samples that you're smelling here. And this is very important for us locally in the Arabian Peninsula. So you may think of stories of Arabia and the frankincense and the myrrh and the disorienting smells that are there. If you ever read Lawrence of Arabia or anything like that, there's always this mention of perfumes as being very, very important. And it's true pretty much all over the Silk Road, but in the Middle East, they use very intensively these incenses called bakhur, which are actually from a tree. And they use it in two ways. They burn it as an incense, just on some charcoal. And the other way is that they use it in a steam distillate, and then they use that in their perfumes. Well, it turns out that this tree only makes these perfumes after about 10 years and after it's been wounded, and it's become endangered. So you cannot buy these in Europe. They're completely on the site's list of endangered species, but they're very popular still in the Middle East. And so together with our team and another professor at KAUST, what we did is we went to the local market and we bought many different samples, and we characterized with gas chromatography. This is what's called two-dimensional gas chromatography. We characterized all of these different samples. We were able to find that there were about 300 unique chemicals in each one of these samples, and each one had maybe about 500 total chemicals that gave it its unique odor. The problem is that we wanted to recreate it, and you can't recreate 300 chemicals with synthetic chemistry very easily. So if any of you are chemists in the room, maybe this will be more interesting to you. So what we did is we took our algal cell and we modified its metabolism to make more of the precursors. You don't need to know the details here, but basically carbon flows through an algal cell the way it wants to. And what we did is we tricked the algal cell into making more of the C15, the 15 carbon precursor that we needed, by changing its pigments and by knocking down another pathway. So in this way, we trick the cell into making this red pigment, the same one I talked about at the start that's in flamingos and shrimp. And by making this red pigment, it sort of turns on all of the precursor pathway in a deregulated way. So it's saying, oh, I'm making a new pigment. I don't know how to control carbon anymore. That gives us more of a pool to work with. And then what we did is we looked at the chemicals that were in this perfume. And we said, oh, there's some base chemicals there that then are functionalized into more complex chemicals. And we can find genes for those base chemicals from different plants. And we can make those base chemicals from our alga. So this gave us a starting point where all of a sudden, instead of having, we want 300 chemicals, right? But what we have here now is an ability to make 9 base terpene chemicals. Each one of these has its own value and different role, but that's not the important part. We then use that process I told you about, the liquid extraction. So we cultured our cells, we have some solvent. We then take the solvent, mix it with ethanol. But then we used another process step where we had a very, very thin membrane, what's called a nanofiltration membrane. And we took all of the ethanol and we put it on that membrane and we pressurize it. And you push the ethanol through the membrane, but all of the perfume molecules stay on the retentate side. So you can concentrate them down. If you tried to do this by like rotovapping, you would lose all of your fragrant chemicals. So this is a very important procedural step. But then all of these terpenes are alkenes, so they have double bonded carbon, right? And what you can do is you can actually attack that double bond with boron and then treat it with hydrogen peroxide, and you can get random chemical functionalization with hydroxyl groups on that chemical backbone. So now all of a sudden we had 9 chemical backbones that were all in various ways hydroxyfunctionalized. And that gave us this lovely perfume that you're smelling right here. So from a base starting point in the alga, we could make a process that made 9 chemicals, and we could make up to 103 different chemicals from that 300 that we wanted. We're not there yet, and the more chemicals you start with in that starting reaction, the more you end up getting in your final product. But this is now published as a preprint, and we're hoping it's accepted in the next week. Actually, we just submitted revisions before I came here. But you can see in the chromatogram on the right-hand side, our terpene mixture has at least some of the chemical complexity that's found in the natural sample. And of course, our alga is no longer green at the start anymore, but brown because we've engineered this red pigment into it. Okay, we've talked about many different things in terms of the chemical engineering space of algae, and we've really only focused on insoluble products from the algal biomass and the secretion of those into a solvent. I just want to talk very, very briefly about how we marry this concept with larger scale production. And you remember that I was saying nitrogen and phosphorus in wastewaters are probably the biggest source of input for algal culture. Well, what we did is we coupled our algal production processes with the effluent that comes from human wastewater treatment. So there's a method of human wastewater treatment called anaerobic membrane bioreactors. And through anaerobiosis, they eat all of the brown stuff in our wastewater, all the organic carbon. The thing is that they emit ammonium and phosphate containing effluent. And you know what? That's perfect algae food. So we married these two processes. We took their effluent, we grew our algal cell that was producing a perfume in it, grew really well under different environmental conditions. We milked that product out of it, and we produced literally perfume from poop water. It's also eau de toilette, if you ever look at a perfume bottle, you know, so think about that next time you go shopping at Chanel or something. So this is how we work with our algal cells. I think I'm probably running out of time, right? Like, how long have I been speaking? Do I have 5 more minutes, max? Okay, good. I'm going to skip over the next topic, which was just the production of volatile isoprene, and I want to tell you about alternative strains to work with for doing biotechnology. And so one of the strains that we work with is much more appropriate for our local environment. A green alga is nice, but you know, it has its limits, and we have very, very hot temperatures where we are. Oh, that didn't change at all, did it? How do I make this change? Uh-oh. Uh-oh. I don't know. I'm just gonna quit and then restart. Hold on. Okay, you can see that. So our environment is very hot, especially in the summertime. We have, you know, temperatures up to 50 degrees sometimes. It's important that we have organisms that work with us as well. So we started working on the engineering of extremophiles, and specifically thermoacidophiles. And there's a couple reasons for this. One, because of the temperature, but two, acidophiles can take a lot of ammonium, so they can take very rich effluents that other organisms cannot. And in addition, the organisms we started working with are actually really easy to handle in the lab, just like the other green algae we work with. So we've focused very specifically on one called Cyanidioschizon marole 10D, which was isolated in Naples in the 90s. This strain is very happy growing at pH 1. That's really acidic. And 50 degrees. That's very hot. So it's growing in hot lemon juice or Coca-Cola, right? You know, if you put your tooth in Coca-Cola, it's gone overnight. It's the same with their culture media. It's full of sulfuric acid. But we've also been able to metabolically engineer this strain to produce different products and grow it outdoors in our extreme environment. I don't have time to go into the details here, but again, we built a bunch of tools, we optimized a transformation protocol, and this is all enabled by the advent in DNA synthesis. So we could design something in a computer, print it, and then put it into an organism and see a phenotype happening. And here we went after the carotenoid profile. So this is the base carotenoids of the organism. And with the expression of 2 different genes, we were able to convert those base carotenoids into different products and turn the green cell into this browny dark red cell. So that just to keep your mind a little bit active as sort of a fun mental game, these are phylogenetically considered red algae, but they've lost the red pigment, so they look green. But we took genes from a green alga and we put them into that red alga to make the green alga look red again. The red-green alga look red. Yeah, try and keep that straight. It also produces a blue pigment that's soluble, so it gets even more fun. But basically, each transgene that we put in had a different effect. You can really see that here in thin layer chromatography. So you don't have to know what's happening here, but basically the wild type strain does not have any red pigment. If you put 1 gene in, you get some intermediate red pigments. If you put 2 genes in, you get this final red pigment. So we were able to pretty much in about 6 months go from concept to phenotype in the alga because of the advent of DNA synthesis and synthetic biology and our ability to handle the organism. And as I mentioned before, soluble, insoluble products, you have carotenoids being insoluble, you have blue phycocyanin being soluble, and then the different engineered products that you can get out of these different strains. So now you have a strain that can grow in conditions that nothing else can grow in and produce these valuable products using pure CO2 as an input because it doesn't care about the acidification. So it's a pretty valuable strain. We're very excited about it. And just to show you that we're putting our energy where our heart is, we're, again, in a very hot environment. And we were able to grow this strain outdoors in August in Saudi Arabia. No problem. It loved it. It was actually too cold for it. It would have preferred hotter temperatures to get to higher biomass. So there's a lot you can do with very little with these strains. Okay, I hope that the next slides work. Yes, I've taken you on a big spectrum of different ideas today. We've gone from finding new species to engineering them to designing bioreactors and a little bit talking about how those resources work in the real world. I hope it was not too complicated, too biologically focused for you. And if you have any interest in any of our publications, this QR code will take you to our lab website and you can download them all there. They're all open source. I pressed the wrong button again. Just like to thank my lab. These are all the nice people I work with, and thank you for your attention.
Bueno, orain, ino galderarik baldin badu, mikrofona pasako ditugu, ados?
Yeah.
Meanwhile, I'll do the first question. So in this case, like in the, sorry, I'm a chemical engineer, so I'm very limited in terms of the genetic modifications, etc. But I'm interested in the liquid extraction process that you carried out. How first you tried with dodecane, and then actually like you moved into like the heavier liquid product. How do you think of using that, or how is like the background? Do you have like chemical engineers? Like, do they like suggest you using that, or where did that come from?
Okay, so it's a fantastic question, and it actually illustrates exactly why you need to be always aware and talking to your boss in the lab. So why did we start working with fluorocarbons? You use fluorocarbons for doing single cell microfluidic droplets. So the cell gets immobilized inside a little vesicle and then can go down a microchannel. And my student accidentally poured too much of it into a liquid culture. And he said, Kyle, the solvent is on the bottom of the culture. And I said, well, that's really interesting because we have this problem with bubbling in the light solvent. Why don't you see if there's any terpenes in it? And so he put it, first he did some research to see if he could put it in the gas chromatograph, and then he did put it in the GC, and then we did see a peak, and then our minds were blown. And then we spent about 6 months really trying many different fluorocarbons. So there are dozens of these that you can buy, and each one has a different boiling point, different viscosity, whatever. And so we just had to try some, and it turns out that there are processes using fluorocarbon gases to extract terpenes from plant matter. So there are patents where you have a plant in a chamber, you inject a fluorocarbon gas, it extracts all of the terpenes, and then you volatilize, you release the pressure, and then the gas goes away, and then you have your terpenes left. But in this case, we don't have to put any pressure on the cells, and it's not evaporating, which is actually really bad because they're horrible greenhouse gas fluorocarbons when they're released, right? Like CFCs. So the liquid extraction nature was very important for us to keep. It turns out that in the 90s, there was a big push for fluorocarbon cell culture combinations because you can highly oxygenate the fluorocarbons. So for human cell culture, you can sort of have an interface where the solvent is giving oxygen to the cells and the medium is on top. But nobody ever did this with terpenes before. So yeah. There's a question back there.
Mr. Aleric?
How is legislation doing around this topic? I mean, can you put a photoreactor at your house to grow a local microalgae and then use it as fertilizer, for example, with compost?
So I think the problem with doing that is not the growing of the algae. That's really easy. It's the downstream processing of the biomass. And I think that regional hubs will have to exist to have downstream processing or have mobile trucks that show up at your house and then harvest your reactor for you, because you need centrifugation or filtration, you need pumps. It's 80% of the cost is in the downstream processing. The legislation of just growing an alga, anybody can do it. There's no problem with that. With anything that's genetically modified, that's a different story. Then you need a proper containment lab.
But if there is not a specific component that you need from the alga and you just want to use the biomass for making the soil much richer in nutrients, could you use, for example, the whole solution with the water without filtrating the algae?
Not really, because you'd need to break the algal cells. Otherwise, you're just putting a bunch of algae on your soil and they will eat all the nitrogen before a plant can. So you have to lyse them. And then algal biomass in general is great, what do you call it, phytostimulant, because of all of the hormones that are released when the cell breaks, breakdown products of carotenoids, phycochelatins. There's a lot of different chemicals that come in algal biomass that make good phytostimulants, but you have to rupture the cell, and that's the limiting factor.
Mr. Aleric?
Question?
So I hope I can phrase my question correctly. So you're manipulating algae like with foreign DNA, right? So is there also a case where you're manipulating algae to be more accessible to their original DNA? Because I know, for example, Batrachospermum, which is like a red freshwater algae, has like slime around it, so it's very hard to work with. Do you do stuff like this as well, or?
In the green alga we work with, that's what I was talking about, is domestication. That was achieved just by blasting it with UV or chemical mutagenesis to then find the phenotype you're looking for. That extracellular mucilage is actually very valuable, so you don't want to get rid of that. And I think it's about figuring out which cell type you want to engineer for what purpose, right? If you wanted to make mucilage variants, maybe you could do it just by randomly mutating it, but actually structurally changing that is very challenging. Nobody's really doing that yet. Okay. Just like starch modifications, like you can do it, but nobody's doing it yet. Yeah, it's domestication. And in general, we have just scratched the surface on algal diversity and their domestication. I mean, you can think about every plant in the world and how different they are. Algae are 5 times more different. They have primary, secondary, tertiary, quaternary endosymbiosis. They're very different organisms. So, yeah, random mutagenesis is where you start. Yeah.
Thank you.
One question. This process can be used to produce any type of biofuel?
Yes, but you don't want to use the GM engineered strain then. So depending on the alga you use, it has different ratios of macronutrients, right? The extreme algae that we grow at pH 1, pH 2, it's 50% protein. It's more protein than a steak per biomass. But, you have to figure out a way to get that protein into a form that somebody wants to eat. Surprisingly, that alga secretes lactate, and it smells like grassy milk when it's growing. So theoretically, you could drink it if you wanted to, but I wouldn't, because it will live at the same pH as your stomach. So probably want to break it up. But yeah, I mean, the addition of algal biomass into human food is happening already. There's different companies making algal biomass as protein alternatives. If you knock out photosynthesis and grow them on sugar, you can get very high densities. And then you can have strains that aren't green anymore, but are heme containing and look red or yellow. And then you can use them as the impossible burger alternatives, for sure.
No, but like biofuel.
Like organic
Like
food. Like,
Yes.
bio, yeah. So as long as your inputs are bio certified, you can treat it as a bio food, right? So if you use a purified chemical, then it's no longer bio, right? But if you use manure and then you strain that manure and you get all the nitrogen and phosphorus out, that's a bio input. Yeah.
Okay, I would like to thank you for the interesting talk and also thank Gorka and the faculty for making it possible. I'm going to like to ask about the species screening. I have been commenting outside with other researchers that almost all the studies you can find in the literature are made with the same 10 or 20 species. And it's very low number comparing with the extremely high diversity we have outside in the nature. Are you making something in this point? Are you making big screening of culture collections, something like this?
So we're trying not to import strains. We're trying to use the ones that we find locally. The thing is, there's no real natural environment in Saudi because people just bring whatever they want. The gardens are all imported species. So we're working with what is adapted locally. The problem is when you start a culture collection and you don't use the right technique, all you isolate is green algae. It's super easy to isolate green algae. They grow really fast. They take over everything. You mentioned that you pipette very specifically cells out of a culture and then grow that. We use an ELISA machine plate washer that can fill a 384 well plate with liquid, and you take your concentrated seawater that has you run 10 liters of seawater through a filter, you keep everything on top, and we put that through that machine and then try and put like 1 cell per well and see what grows back. But again, we're biasing for whatever grows. And I'm okay with that because as a biotechnologist, we only care about things that we can grow. We don't really care about the fundamental phycology, but it's important to do both. And so we're just growing everything we possibly can at this point.
Beste galderarik? Ez? Bueno, nik uste ja denboraz nahiko luzatu garenez. First, I would like to thank you for coming and for doing this such an interesting presentation that, at least in my case, involves so many different aspects that I think that it's interesting for our faculty that has all these degrees that are taught in the different fields, and especially thank you for making it easy for us to have you here too. You are very, very busy. You have so many things to do, and well, at least in my case, I'm very, very happy of how the thing went.
Thank you very much for having me.
Eta eskerrik asko zuei ere etortzeagatik eta parte hartzeagatik.